The physical-chemical properties of the virus in crude sap, such as dilution endpoint, thermal inactivation point, and longevity in vitro, would provide useful data on the concentration of the virus in the living host and its stability that would help in the transmission and virus purification experiments. Although these properties are not being used at present to identify and characterize a plant virus, they are quite useful in getting an idea about the concentration and storage duration of a virus in the active state in crude sap for future courses of study. However, you can exclude suspected viruses by their properties. For example, viruses with high stability like tomato brown rugose fruit virus, TBRFE, from Tabama viruses has high dilution point. On the other hand, tomato spotted wilt virus, TSWV, from Tospo viruses has a low dilution end point, and they both have similar symptoms. So if the virus infected your crop has low dilution end point, it will not be TBRFE from Tabama viruses, and the causal agent could be TSWV from Tospo viruses. Material and tools used in our experiment. 1. Tomato plant infected with TBRFE. 2. 22. Healthy tomato plants. 3. Mortar and pestle. 4. Abrasive material, carborundum. 5. Labels. 6. Test tubes in a tube rack. 7. Aid pipette. 8. Potassium phosphate buffer. 9. Cheesecloth. 10. Stirrer distilled water. 11. Thermometer. 12. Water bath. 13. Ice bath. 14. Bottle. 15. Beaker. 16. Pipettes. Dilution endpoint, DP. The dilution endpoint, DP, is the highest dilution of sap from a virus-infected plant which is still infectious, but is usually given as the range between this dilution and the next one at which the infectivity is lost. 1. Label as follows. 8 plants. 2 plants for 10:1 dilution. 2 plants for 10:2 dilution. 2 plants for 10:3 dilution. 2 plants for 10:4 dilution. 4 test tubes. 10:1. 10:2. 10:3. 10:4. 2 add 8 ml of potassium phosphate buffer to the mortar. 3 cut the symptomatic leaves of the infected plant into small parts and put them in the mortar, then grind them excessively. 4 squeeze the mixture through a cheesecloth. 5 fill the test tubes with 9 ml of stirrer distilled water. 6 add 1 ml from the crude sap to the first tube, 10:1, by pipette, then shake the tube to mix it. 7 add 1 ml from the first tube, 10:1, to the next tube, 10:2, with a new pipette. 1 ml from the 10:2 tube to the 10:3 tube, and 1 ml from the 10:3 tube to the 10:4 tube respectively, then shake the tubes to mix them. 8 apply carborundum on the plants. 9 inoculate 2 plants for each dilution mechanically. 10 place the plants in the greenhouse. Below is our records of the observation of the symptoms. All records were the same for all plants inoculated with inoculum in different dilutions. First week, no symptoms were observed. Second week, no significant symptoms were observed. Yellowing is mostly due to nitrogen deficiency and low light intensity. Fourth week, mild mosaic. The red color is due to the cold weather. Fifth week, filiform, narrowing, small leaves, and mild mosaic to yellowing. Discussion. Dilution endpoint DEP, tomato brown rugose fruit virus, TOBRF, remains infectious with the same severity for all dilutions, 1 × 10⁻⁻⁻, 1 × 10⁻², 1 × 10⁻³ 1 × 10⁻⁴, which means that the DEP is more than 1 × 10⁻⁴, and this refers to the high stability of the Tobamo viruses. Longevity in vitro, LIV. Longevity in vitro, LIV, is the length of time after which crude sap from a virus-infected plant loses its infectivity when kept at room temperature. 1. Label as follows. 6 plants. Bottle. 2. Add 8 ml of potassium phosphate buffer to the mortar. 3. Cut the symptomatic leaves of the infected plant into small parts and put them in the mortar, then grind them excessively. 4. Squeeze the mixture through a cheesecloth. 5. Apply carborundum on the plants. 6. Inoculate the plant on the first day mechanically. 7. Fill the sap inside the bottle. 8. Inoculate plants each day for another 5 days, total equals 6 plants in 6 days, and place them in the greenhouse. 9. Check the plants for the next weeks to observe the symptoms. Below is our records of the observation of the symptoms. All records were the same for all plants inoculated in different days. First week. No symptoms were observed. Second week. No significant symptoms were observed. Yellowing is mostly due to nitrogen deficiency and low light intensity. Fourth week. Shoestring and stunting. Fifth week. Shoestring and stunting. Discussion. Longevity in vitro live, tomato brown rugose fruit virus, to BRFE, remains infectious with the same severity for all days, 1st, 2nd, 3rd, 4th, 5th, 6th. Which means that the live is more than 6 days, and this also refers to the high stability of the tobamoviruses. Thermal inactivation period, TIP. The thermal inactivation point, TIP, is defined as the lowest temperature required for complete inactivation of a virus in crude sap heated for 10 minutes. Label as follows. 8 plants. 4 test tubes. 2. Add 5 ml of potassium phosphate buffer to the mortar. 3. Cut the symptomatic leaves of the infected plant into small parts and put them in the mortar, then grind them excessively. 4. Squeeze the mixture through a cheesecloth. 5. Add 1 ml from the crude sap for each tube. 6. Install the thermometer inside the tubes. 7. Expose the test tubes to the temperatures using the water bath for 10 minutes. 8. Submerge the tubes immediately in the ice bath. 9. Apply carborundum on the plants. 10. Inoculate 2 plants for each temperature mechanically. 11. Place the plants in the greenhouse. 12. Check the plants for the next weeks to observe the symptoms. Below is our records of the observation of the symptoms. All records were the same for all plants inoculated with inoculum exposed to different temperatures. First week. No symptoms were observed. Third week, leaves are filiform. Fourth week. Sever leaves filiform and stunting. Discussion. Thermal inactivation point, TAV. Tomato brown rugose fruit virus, to BRFE, remains infectious with the same severity for all temperatures, 40 Co, 50 Co, 60 Co, 70 Co, which means that the TIV is more than 70 Co, and this refers to the high stability of the tobamoviruses.